Hello, good evening, and thanks for coming to tonight's Life Without Disease lecture. My name is Mitchell Cronenberg, and I'm the president of the La Jolla Institute. So we've had a series of these lectures that bring our work, we hope, to the public so that you can have greater understanding and appreciation for what we're doing. Usually we do them at the Institute, and tonight we decided to have it here at the Sanford Consortium for Regenerative Medicine. It's a collaboration between 5 organizations, and we are happy to be one of the 5, along with UCSD, Sanford Burnham Medical Research Institute, Scripps Research Institute, and the Salk Institute. So it's a wonderful organization, people working on various aspects of stem cell biology from all over the Mesa. So it was an experiment to have the lecture here tonight instead of in our building. For your regular attendees, it gives you a chance to see something else. And we didn't count on the rain, but that's how experiments go. So our speaker tonight is Sujan Shresta, and before she speaks, I'm going to have our Executive Vice President, Steve Wilson, give you a little bit of background so everybody can have the same level of understanding of immunobiology. Steve is a longtime colleague of mine, very valued member of our institute. He has a PhD. He was very interested in information technology, actually has many interests, but he decided to move into our information technology department and to run that. And eventually he's become the chief administrative officer of the institute. But he also participates in a number of our scientific enterprises, especially a national database on what's called epitopes, that are parts of viruses and bacteria that your immune system recognizes. And Steve is an active participant in that and in other scientific endeavors at the Institute. So without any further delay, Steve, come up. Thank you.
Thanks, Mitch. Well, thanks for braving the San Diego downpour. Growing up in Chicago, I was excited to use my umbrella, not just to block the sun, but to block the rain. We're going to talk tonight about combating infectious disease, and really on a global level. But before we do that, and before Dr. Shresta comes up and really gets into the guts of it, I'd like to pose a couple of questions and answer them myself. I'm sure I'm going to get them right, but to really acclimate us all to why it is that we do it at La Jolla Institute. So one of the questions that would be worthwhile asking is in the modern world, in the very recent history, where we look at how great microbes and the interaction of microbes are, how can we still be fumbling over or worried about bad microbes coming in and getting us? You know, don't we know enough about these things to really do something about it? In fact, our microbiome and the viruses and bacteria that live in and on us are critical to our health. And so the eradication of the microbiome is in there. But really it's become kind of clear as we look at the human condition that we strike a balance. And so maybe it's an easy question. You know, if we're all interested in good microbes or the balance, what's going on? And what's going on is when microorganisms infect us and dysbiosis, the incorrect balance of microbes in us is there. And there's out of control growth and what we commonly call sickness and infection. And I bring this up because the very concept can be kind of interesting if you look at what's going on. We're all bathed in microbes. Our bodies are exposed to them constantly, and we've evolved to do that. But things can actually run out of control in a couple of ways that become relevant to when we as advanced humans now want to control it again. So first off, microbes are small. They're so small you can't squish them. They'd fit right in between the crevices of your skin cells. And when I say small, I'd like to put some scale into perspective here. First off, if there are in your body some 10 trillion cells, there's another 1 to 200 million non-human cells in your body. And they all fit because they're small. So kind of counting by tens, you can look at the size of just a molecule or a cell membrane. Antibodies, which we're going to hear about, which are produced by your immune system. And then viruses, and this is length here, so about 100 nanometers. And because bacterial cells and animal cells are so much bigger, viruses actually have evolved to live inside and take advantage of the host for their biology. So right off the bat, you can imagine when you start counting who's what and where are these viruses, for instance, you have to start looking inside of cells to see them as well as outside. But also, viruses of about that size, you can put 1 million of them in a human cell, which means they have a size advantage, not in terms of their actual size, but in the total number and what they're up to. And they co-opt what we're doing, okay? But no surprise, we have an immune system that can see and destroy microbes. So tonight we're going to talk about what goes bad, why isn't the perfect balance always there, and why when these foreign invaders come in, doesn't your immune system do what it does most of the time and get things under control? Well, some of the reasons are there can be overgrowth. They hide, they change shape, and every once in a while they actually pick the wrong cell to invade, and when they do, things tend to go off the rails and really bad outcomes come. So maybe viruses have good intentions, but when they get into the wrong place, they can cause tons and tons of havoc. One of the things that they do in particular is they can change the way your immune system acts. Now, we don't have enough time to go over the immune system again, but there are parts of your immune system that respond quickly, the innate, and those that develop memory, adaptive. And this is just a large diagram of all the different ways that viruses over time have interacted with our immune system and escape potent immunity. In other words, our immune system launching in and specifically destroying them. And this happens to be hepatitis C. But viruses, the interesting takeaway here is viruses really take a look and have co-evolved to interact with our human immune system in specific ways to dampen down the removal of them so that they can survive and replicate. And of course, we haven't just sat still about it. We've developed antiviral drugs. So we turn around and we try and target the life cycle using pharmaceuticals to target things that viruses do. And a question might be, well, why not just make lots of this? Tons and tons of antivirals. And the biggest reason is because when you try and attack something that's inside your own cell and has used your cell to produce itself, in a way you have to attack your own systems. And so many antiviral drugs have toxicities to ourselves. This is unlike many times with bacteria where you can take antibiotics and just clean out the bacteria. With antivirals, there tend to be a lot of side effects. Nonetheless, we've made a lot of them. But there still aren't enough because different viruses do different things, and it's a challenge. So, why not make loads of vaccines against all the different viruses? In 1951, that was good enough to get a Nobel Prize. This is a very grim-looking Max Thieler here, but he got a Nobel Prize for inventing an attenuated vaccine for yellow fever, easily one of the most effective vaccines out there. Hundreds of millions of people are protected by it. It's extraordinarily protective over your lifetime. But it turns out, and this is what Sujan's going to talk about, making vaccines by simply getting a weakened version of a virus and putting it into a shot, or taking pieces and putting it into a shot, doesn't always work. As a matter of fact, many times it doesn't work. And it's complicated, but it comes down to how your immune system sees it. And so again, striking the balance with these microbes that we know that we have some reliance on really comes down to being able to take out just a few very specifically without messing with anything else. So before I introduce Dr. Shresta, I also need to get off my high horse about how viruses are just bad, bad, bad. A couple weeks ago, there was a great report in Nature, a geek journal for immunologists and all scientists, about how viruses come in, interact with the lumina of your intestines, the cells in your intestines, and can actually restore some function that is lost when you don't have the right bacterial component. So this is a very interesting clockwork that we're all getting into here as we look at the immune system, how it balances the good virus, the so-called good viruses, the bad ones, how your immune system chooses not to attack some because it may be needed. It's all very, very interesting. So with that, I don't want to cut into Sujan's talk. She's going to talk more, but before she does, Dr. Shresta is a principal investigator and professor in a lab at the Institute. Her lab is in the division now of inflammation biology, but she's a virologist. You can read her bio. What you don't see in the little sidebar after Smith College, WashU, and Berkeley are the high honors she gained at all of these institutions. She was awarded a Blasker Award, and in just the few short years she's been at the Institute, has really put together an amazing program in infectious disease, and in particular dengue fever. So without further ado, Sujan.
Thank you, Steve. Can everybody hear me? Okay. Thank you, Steve, for the generous introduction. Thank you to all for attending here, especially in this rain. I'm happy to be here and have this opportunity to talk to you about our work on this viral infectious disease called dengue hemorrhagic fever. Not many in the US know about dengue hemorrhagic fever, but as shown in this slide here, unlike dengue hemorrhagic fever, now, as of this summer, many of us know about this virus, Ebola, which also causes hemorrhagic fever. So in this slide, and the next couple of slides, I'm going to talk about Ebola to highlight some of the key issues surrounding the challenges associated with combating these infectious diseases that are very important at a global level. The Ebola outbreak started in West Africa in these three countries, Sierra Leone, Guinea, and Liberia. And as highlighted by the Dallas case, where this person from Liberia was visiting relatives in Dallas, he was the first patient that was diagnosed with Ebola in the US, and he went on to die, but in the meantime, while caring for this patient, 2 nurses were infected, but they recovered. So these 3 Dallas cases highlight the fact that infectious diseases such as Ebola really have no geographic or economic barriers. So, as you know, the current Ebola crisis is still ongoing. In the best case scenario, people have estimated in this country, the Liberia, which is one of the hardest hit countries, this is the hardest hit country. The best case scenario is that by mid-January 2015, we'll have anywhere between 11,000 to 27,000 cases of Ebola. The worst case scenario, by the same estimates, is we could have anywhere from over 500,000 to over almost 1.4 million cases of Ebola. So imagine, what if we had a vaccine right now, which, as you know, we don't. Then we won't have to worry about this worst case scenario. We can focus more on these types of situations. So another point that I would like to make with regards to Ebola is, here I'm showing you the number of human deaths in red. So Ebola was first recognized back in 1977, and small outbreaks have occurred throughout. And now in the blue are the number of publications. So that is a gauge of how much research, scientific research, has been going on to understand how this virus is causing disease and how we can prevent infection and develop vaccines against this virus. We see that not much has happened in all the several decades after this first Ebola outbreak. After each little outbreak, yes, we have a little spike, and of course there's now this massive Ebola crisis going on. But still, this is not a very many numbers of scientific publications. So those 3 slides about Ebola, I put together to highlight the following 3 points. First, as I said in the first slide, these infectious diseases have no geographic boundaries. Second, as highlighted by my second slide about how research is happening after the Ebola event, Ebola outbreaks, that's not helping us. We need the vaccine now. And all this basic scientific investigation needs to happen before you have these outbreaks. And these outbreaks turn into epidemics. And also with Ebola, what's happening is there is now a massive spike in funding, but that's following the media coverage. What should really be happening is all this research should be happening way prior, several decades in advance. And this way, if there is an Ebola outbreak, we wouldn't be dealing with this massive humanitarian crisis. So with that, I would like to introduce you to another infectious disease that I think is very important and not many in the US know about. It's a mouthful to say. It's called chikungunya. And chikungunya is a Swahili word. It means that which bends up. So it's literally the patients walk like this. It's because of the hallmarks of chikungunya infection is they have massive and extremely severe muscle and joint pains. And often the peripheral joints, such as the hands, the ankles, and the feet, they're both swollen and extremely painful. So this is a two-stage disease. The acute stage is what I said is that which bends up, the stooped posture of the patient. And unfortunate episode of this chikungunya infection is that most of these people, over 90%, go on to have this chronic phase, in which people up to as long as 5 years after initial infection can have rheumatoid arthritis-like syndrome. And as you can imagine, some of these patients, they have quite a debilitating lifestyle for many years after the first infection. Another reason I'm bringing up chikungunya here is because to highlight this nature of infectious diseases in that they tend to have very explosive outbreaks. And chikungunya really highlights this aspect. Before October 2013, the only countries that had chikungunya virus were in Africa and in Asia. So the dark green represents the endemic countries. In October 2013, the first acquired cases of chikungunya were reported in the Caribbean, in the St. Martins. And in 1 year, by November 2014, over 30 countries in the Americas now have this virus, including the US. Almost 900,000 cases of chikungunya have been suspected, and out of those, over 16,000 are confirmed chikungunya virus infections. So this is all happening within 1 year. This virus was not present until this last year in the Americas. And yes, every single state in the US has actually at least the imported cases of chikungunya. So despite this, again, this global reach of this virus, just like Ebola, we don't have vaccines or antivirals to either treat or prevent this virus infection. So another reason I brought up chikungunya is because now it allows me to introduce my favorite virus, or the virus that I work on. It's called the Dengue virus. And what's really interesting about this relationship between chikungunya and dengue is that both are transmitted by the same mosquitoes. So the name of these mosquitoes is called the Aedes mosquitoes, and another nickname some of you may know is called the Asian tiger mosquitoes because of the stripes. And some of these mosquitoes are present all throughout the southeastern United States. So let me tell you about this virus called the dengue virus. It causes a spectrum of illnesses in humans, ranging from dengue fever, which is the mild form of this virus infection. It's also known as break-bone fever because of high fever and severe headache, joint, and muscle pain. The next form of this virus infection is called dengue hemorrhagic fever. In terms of hemorrhagic manifestations, they include bleeding in the eye, nose, and gum. The other hallmarks of dengue hemorrhagic fever is low platelet count, elevated hematocrit, and this this phenomenon called plasma leakage, in which you have leakage of fluid from blood vessels into tissues. If this plasma leakage syndrome becomes severe enough, then patients undergo shock. At this stage, it is called dengue shock syndrome. This dengue shock syndrome, often you have massive intestinal hemorrhage, and yes, this can be fatal. Shown here is the geographic distribution of these dengue-transmitting mosquitoes. These are the tiger mosquitoes in the blue. And the red represent areas that have both the mosquitoes and the virus. You can see that virtually almost all subtropical and tropical regions of the world are affected with this virus. And for us, as I said before, these mosquitoes are now in the southeastern part of the United States. And it's not just a mosquito. As of 2012, July 2012, dengue is now endemic in Florida. So now the US has dengue. So in terms of dengue as a problem for the world, we in the field often like to show this pyramid diagram. 3.6 billion people, that's half the world population, are at risk of infection with this virus. Out of those 3.6 billion people that are at risk, 390 million infections occur each year. Out of those 390 million infections, only about 1 million go on to develop the severe disease. So what's really interesting about this virus infection is that you have this massive number of infections. Most of them are either the mild disease, the dengue fever, or subclinical. Some of them will not have any symptoms. But there's a fraction that go on to develop, it's still a lot, 1 million every year, of severe disease. And out of those 1 million, about 24,000 die each year. Just like with Ebola, just like with chikungunya, despite this global morbidity and mortality associated with this virus infection, we don't have any vaccines or antivirals. So in terms of dengue vaccine development efforts, dengue poses, as Steve alluded to you before, some unique challenges. And the main reason is that, even though I'm telling you always dengue, there are really 4 different viruses. We call them serotypes. These dengue 1, 2, 3, 4, they're called dengue 1, 2, 3, 4. They're only about 60 to 67% identical at the amino acid level. So you can think of them as related, but yet different viruses. In fact, they are more similar to some of the viruses you've probably heard about, the West Nile virus, the Japanese encephalitis virus, than to each other. Based on number of clinical epidemiologic studies, we think, the researchers in the field think, we're not sure yet, but we think that there's a lifetime immunity following infection with one serotype. Some of the virologists in the field may challenge that, but the general consensus is, looks like there is a lifetime immunity following infection with one serotype. But because of the existence of these 4 different serotypes, or 4 different viruses, you could have 2nd, 3rd, and potentially 4 infections. Dengue is really, truly an emerging and re-emerging disease. In 1970, there were very few countries that have dengue, and most of these countries do not have all 4 serotypes. There are 4 viruses co-circulating. Now, in 2004, by 2004, you can see many more countries that have dengue, and on top of that, most of these countries now have all 4 serotypes co-circulating. So remember, infection with one serotype does not give you protection against reinfection with a different serotype. So this co-circulation is not good for the control of this virus infection. So the next question is, why is dengue becoming a problem? Why is this an emerging disease? And the answer is really us. And I'm showing you pictures of all the human activities. That includes massive urbanization, There's, in many of these dengue endemic countries, you have migration of people from the villages, rural areas to the cities. And of course, our lifestyle there, where these dengue-transmitting mosquitoes, they thrive in environments in these kinds of containers with still water. So our use of non-biodegradable containers and tires and flower pots, those are prime areas where these dengue-transmitting mosquitoes live. Other human activities include, really in many of these countries where the resources are very limited, there's a massive breakdown of programs that used to be there in terms of vector control and those public health infrastructure. And of course, the plane, this is massive increased global movement of both us, the humans, and the goods. So I'm bringing this pyramid picture here again to remind you that out of these 390 million infections, only a fraction of them developed a severe disease. So one of the key questions in the field is, why is this? Why are only a fraction of the people are coming down with the severe disease? Variety of risk factors have been associated with why some people come down with severe disease and some with mild disease. One of them includes the race. So for example, Caucasians and Asians are more susceptible to coming down with the severe disease than people with the African genetic background. So the race, the host genetic factors, and age, children tend to come down with severe disease more than the adults. And in certain developing countries, looks like the nutritional status may also play a role. So in this case, people from well-to-do families who are well-nourished, their children don't come down, they tend to come down with severe disease more often than the ones from who are not adequately nourished. And the females tend to come down with the severe disease more than the males. And in the case of infants, this is a very special scenario where these infants that are born to dengue immune mothers, what that means is that these infants have dengue-specific maternal antibodies when they were born. They are also more susceptible to developing the severe disease than those infants that are born to dengue-naive mothers. But of all these risk factors, the single greatest risk factor for coming down with severe form of dengue disease is the secondary infection with a different serotype. So a central question in the field is, why are these people with secondary infection with a different serotype? And in the case of infants, which have these dengue-specific maternal antibodies because they were born to dengue immune mothers, why are they coming down with severe disease? Many hypotheses have been postulated to try to answer this question, but a dominant one is called antibody-dependent enhancement. Sometimes I will say the word ADE, that stands for antibody-dependent enhancement. This hypothesis was proposed almost half a century ago. And basically what this hypothesis states is that the antibody response to the first infection, in the case of these people with secondary infection, or in the case of infants, the maternal dengue-specific antibodies are the ones that are causing severe forms of the disease. So it's really the immune system that's responsible for causing the disease. So this hypothesis is about the role of antibody in causing severe disease, and so dengue hemorrhagic fever is known as an immunopathogenic disease. And the support for this hypothesis came only recently by our laboratory in 2010. So this hypothesis was proposed a long time ago, but it took this long to provide any support. And here I'm showing you the different infection scenarios, assuming that this hypothesis is correct. So in the context of the antibody response to this virus infection. During primary infection, by definition, you're not infected the second time. You develop a nice antibody response, and during primary infection, most people either have no disease or mild disease. During secondary infection, so the circle, this is the virus. During secondary infection with the same serotype, so homologous means the same serotype, so the red virus, the the antibody response to the first infection will neutralize, will recognize the virus and get rid of this virus infection. So this is a nice neutralizing antibody response, so that person will be protected. Now, I'm telling you about the secondary infection with a different serotype, so heterologous serotype. So instead of the red and red virus here, I have the first infection is one serotype of the red virus. The purple is the second infection. It's a different serotype. Not all of the antibody response to that first serotype, this first infection, will recognize and bind this second serotype. This will lead to what we call a subneutralizing antibody condition. That means the antibody response is not good enough to neutralize and get rid of this virus infection. And this is how we get this ADE, this antibody-dependent enhancement-mediated disease. So besides the secondary infection patients, I also told you about these infants that are born to dengue immune mothers. They have these dengue-specific maternal antibodies. And in the case of infants, it doesn't really matter whether it's primary, secondary infection. Somehow, these maternal antibodies that's circulating in these infants is not, again, good enough to neutralize. Instead, they're subneutralizing, and that's causing the severe form of the disease. So this is how we're viewing the antibody response to this virus infection in all these different infection settings. So you can see in this setting, the antibody response is good. Whereas in these two settings, the same antibody response, but maybe the different quality. This is people in our field are trying to understand what are these differences between this antibody response and that. That's allowing these people, that's making these people come down with the severe form of the disease. So back in 2010, how did we provide support for this hypothesis, this role of antibody in these two contexts? And the answer to that question is we developed a mouse model. Specifically, we took a mouse, we injected anti-dengue antibody or immune serum, so the antibodies are in this immune serum, followed by challenge of these mice with the virus. The mouse starts to get ruffled fur. As the infection progresses, we have increased viral load in a variety of tissues in these infected mice. This is what's happened. This is what's happening in people as well. And as the infection progresses, you have a variety of these immune molecules called cytokines. And these are the same kind of immune molecules, the cytokines that you find in people with severe disease that are also elevated, that are found in this mouse. And as the infection progresses, after you have this elevation of these immune molecules, you have all these features that are found in people. That includes elevated hematocrit, low platelet count, this hallmark phenomenon called vascular leakage, increased vascular permeability, and the GI hemorrhage. And ultimately, the mouse dies. So what this work showed is this antibody. Remember, all this is happening because we injected this antibody into this mouse. This phenomenon called antibody-dependent enhancement converts a mild disease into this severe lethal one. In the absence of this antibody injection, this mouse would go on to live. So the only difference in life versus death and turning on all these immunopathogenic mechanisms in this mouse is the injection of this antibody. So you can imagine now this phenomenon called antibody under certain conditions, especially in the vaccination settings, may be a problem because most of the vaccines that we have now are associated with eliciting the antibody response to that pathogen. And if we employ that same approach, you can imagine this may not be the best approach in solving the dengue vaccination problem. Another value of our work, besides demonstrating ADE, is that now this is the first time we have a relevant animal model. This is the only animal model that shows all the hallmarks of the human disease shown in the blue that I've told you before. And in the next slide, I'm going to tell you about how we're using this mouse model that we developed in 2010 to address some of the questions related to the safety and efficacy of developing dengue vaccines. So in the next slide, I'm just going to summarize our work. What we do is we inject a mouse with the virus, And this is the baseline levels of virus we find in the mouse. So this is a control non-vaccinated group. Another control that we always use in these vaccination experiments because of this phenomenon of ADE is we inject these mice with anti-dengue, these dengue-specific antibodies, so that they will enhance, so at some neutralizing condition, and then challenge the mouse, and we know that with this protocol, the mouse will come down with this ADE-mediated disease. They have high viral load, and they have this ADE-mediated disease. What I'm summarizing here now is our work with inactivated vaccine. So some of the vaccines that we have now they're basically dead version of the real pathogen. So we used one of our inactivated virus as a model vaccine, injected that into the mouse, and when we challenged those vaccinated animals, we found that they came down with severe disease. Okay, so and in that work, we showed that it's the vaccine-induced antibody response that was causing ADE. Before, we were artificially mediating ADE by injecting antibody to this mouse, but now, here we have this under certain conditions, as in this inactivated vaccine, you can have severe disease. So how can we, well, this is not good news in terms of dengue vaccine development. How can we solve this problem? An approach we took, we found along the way, was that if we vaccinate the animals with this inactivated vaccine, and then give them dengue immune T cells right after vaccination, just before challenge, the mouse is protected. So in this similar way, we use another vaccine, and this is a different model vaccine. It's a subunit vaccine where the vaccine is eliciting an immune response against only a portion of the virus. So in this, so some of the vaccines that we currently take are subunit vaccines. So when we injected this mouse with this subunit vaccine and challenged them with this lethal viral challenge dose, we found that the mice were highly protected. In these mice, we couldn't find any virus. So now, not only is this a great vaccine, and we should try to develop this kind of vaccine for people, in terms of basic investigation in my laboratory, we have a model to figure out how this vaccine is mediating protection. And what we found was that, answering this question, how is this vaccine working, is that when we transferred the vaccine-induced antibody response into a new mouse and challenged that mouse, we found that even though the vaccine is protective, the antibody response to this vaccine is enhancing. It's bad. It causes ADE. However, when we transferred this vaccine-induced T cells, we found that the mouse is protected. So basically, this vaccine is working by eliciting T cell response, and this is the T cell response to this vaccine that's causing protection. So based on this type of work, we feel that for a successful dengue vaccine, we should focus our efforts to maximize both safety and efficacy. We should focus our efforts to elicit both the antibody response and the T cell response. And in terms of the cellular response, based on our work, it should be both CD4 and CD8 positive T cells. Besides vaccines, my laboratory also works on testing antivirals. So the value of this mouse model is we can, besides testing vaccines and trying to figure out how the vaccine protects, or in certain cases enhances the disease, we can also try to identify candidates that can be used to treat people and look at the safety and the efficacy, just like with vaccines, and how these therapeutics work, these candidates work. So one of those drugs, so we have collaborations with many partners, both in industry and academia. And one of our partners, called United Therapeutics, their subsidiary is United Virology. Based on our data, they are now in phase 1 trial. So this is a pretty exciting result for us, that their drug candidate, which targets the host process instead of the virus itself, is able to prevent severe disease in our mouse model. So based on that data, they're now going into phase 1 clinical trial. So in terms of the future of these important infectious diseases and what I think will be happening in the next few years, due to this current Ebola crisis, I think we will have the first Ebola vaccine in the near term, in the next 2 to 3 years. This vaccine trials have been fast-tracked at an unprecedented level. And recently, Sanofi reported the phase 3 trials of the first ever dengue vaccine. And looks like the results they're not great, but they're good enough so that they will be licensed. And I think because of this current chikungunya epidemic, the chikungunya vaccine program will be fast-tracked, and we will have the vaccine against, at least one of the first vaccines against chikungunya in the next 5 years. After that, I'm optimistic about some of the drugs that we're helping push through in the clinical trials. And so we should have some first line of drugs against dengue and for sure against Ebola. And in the next 10 to 20 years, we should have the next generation dengue vaccines that we hope work better than the current first generation dengue vaccines. And the same thing with the antivirals against both dengue and chikungunya. So with that, I thank you for your attention, and I'm happy to take questions.
Before opening it up to questions, I'd like to thank Sujan for making a great presentation. Her work is of tremendous interest to a number of biotech companies that are trying to develop antivirals, as well as being of fundamental interest for understanding this important infection. I'd also like to thank all of you for coming once again. We're very grateful for your presence here. I believe our organization is very special. It is a independent research institute, a very efficient and collaborative organization, but one that is focused on immunology and the immune system. And we believe that studies of immunology and the immune system really can have an enormous impact on health. And I think the talk today really very clearly illustrates that. So we're mostly supported by the United States government through the National Institutes of Health, through a very competitive granting process. And funding has not been growing, as I'm sure many of you are aware. So we're very dependent upon supporters like you who are interested in our work and who are willing to make contributions or to connect us to those who can make contributions. So I'd like you to think about that. Or alternatively, if you are members of organizations, we're very happy to go out into the community. Getting our name out is very important. And we welcome having more contact with the public. So after we have our question period, there will be a reception. It's downstairs in the lobby of the Sanford Consortium for Regenerative Medicine, and I'll be there, Steve Wilson, perhaps some of our other faculty, of course Sujan will be there, and I really invite you to continue the discussion there, and there'll be some refreshments as well. So now, without any delay further, any questions from the audience? I have one actually, but I don't want to ask the first one. You're good, go ahead. Yeah, there's a question.
The funding for this whole thing, it seems like it's a worldwide endeavor that needs to be started on or increased. What's happening with that? Is there a worldwide organization that can produce?
So Bill and Melinda, the Gates Foundation, they used to support some dengue research, but at the late stage level, where mostly for helping the companies do the vaccine trial. So it was not really for basic scientific investigation. And in a number of the countries, yes, it's really the government programs in countries like Singapore and Thailand, but they can't match the kind of scientific powerhouse that the countries like the US and Western Europe have. And just like many in the US don't know about dengue hemorrhagic fever, people in Western Europe also don't know about this virus.
What about the World Health Organization?
They don't have the budget. Right now, everything is going to Ebola. Before that, it was HIV and TB and malaria, and there's just not enough resources. And as I highlighted with my slides at the beginning, what will probably happen is there will be a massive dengue outbreak and then a spike in funding, and it'll be a little too late.
The four different types of dengue, is your protocol, does it confer resistance? How many types are there for mice, or is it exactly the same virus?
So the virus that we use to challenge these mice, they are still dengue, but one of the main reasons people have not been able to confirm this hypothesis is because mouse is not a natural host. This virus infects only mosquitoes and humans, not even primates. So not even primates get any of these disease symptoms. So my lab's contribution to the field is really making what we call these mouse-adapted viruses that are different from the human virus only in 1 or 2 amino acids. So that allows the virus to replicate itself in the mouse. And so that, we have to use these special viruses to challenge this. But in terms of the antibody response, the antibodies that we inject to give this ADE-mediated disease, it can be any antibody. Even the human antibodies will work. That's a good question.
I wanted to know how rapidly are these viruses evolving? Some viruses like flu, of course, change every year, and HIV changes in each person infected, of course. So how rapidly are they evolving? And actually, why isn't antibody-dependent enhancement found in other infections? Or is it found and we don't know about it yet?
So I think there's something special about this 60 to 67% identity between these 4 serotypes, between these 4 viruses. They're close. It looks like they're close enough, and they're different enough perfectly, so that the antibody response under certain conditions will recognize and neutralize, and we'll still recognize and we'll do the opposite. We'll mediate enhancement. So I think it has to do with this related levels of relatedness. Whereas with other viruses, with HIV, yes, you have these are all RNA viruses. So one thing interesting I found out while I was researching for this talk was that all these viral hemorrhagic fevers, these are all RNA viruses. And these RNA viruses, what's special about them is, as Mitch was alluding, they evolve. There is no such thing as one viral strain. When people talk about the dengue serotype 1, you have multiple genotypes that's known, and who knows how many unknown variants of the virus. Because one of the enzymes that's used to replicate the virus is very error-prone, and that's actually good for the virus, so that they're always changing. So that anytime the immune system, whether it's the antibody response or the Tcell response, if that antibody or Tcell response is able to get rid of that population, there is a variant that can mutate and now evade that antibody or Tcell response. So tackling these RNA viruses is very, very difficult. And like I said, all these big ones, these viral hemorrhagic fevers, these are all RNA viruses. And in terms of dengue, so how much they evolve, it depends on the virus. For dengue, it's not as fast as HIV, but it does mutate. And then one of the reasons, perhaps the Sanofi vaccine, the results were not that great. It may be that the vaccine was generated using viruses that were circulating back in the 60s, and the viruses have changed enough from the 60s to now. Maybe that's the reason the antibody response to those old viruses don't work in these new viruses anymore. So I think in terms of both vaccine and antiviral development, we do need to think about how these viruses are changing over time. And yes, they do change. Yes?
If the subunit vaccine is hitting the different serotypes in the dengue, that's what it looks like is happening to create a larger response that works. Why do you need the T-cell mediated, how does that come into it? Why doesn't it work just by giving a subunit vaccine and that works every time?
So the response to that subunit vaccine is you have both an antibody and T-cell response to that subunit vaccine. And when people are formulating dengue vaccine, you always have 4 different either subunit vaccines or 4 different live attenuated. You're giving 4 different things, 4 different particles, 4 different live attenuated viruses, or 4 different inactivated viruses. You need dengue 1, 2, 3, 4. So in terms of the antibody versus T-cell response, the reason we need T-cell response is our work is showing that the antibody response, the way we have immunized these mice, or the way these vaccines work in this mouse model, it's not good enough to neutralize, to get rid of the virus and prevent infection. And now, there's a reason why we have 2 arms of the immune system. Maybe it's because of pathogens like dengue. So the host knows, okay, antibody's not working, but you still have another arm of the immune system, the T cells. And so far, all our work is suggesting that these T cells are so good that even when you have this bad antibody response going on, they can override it and still protect you. So I think by evolutionarily, we're designed to have these 2 arms of the immune system so that we can deal with a variety of pathogens in a variety of settings. In the case of the same pathogen, but sometimes the antibody response is good and sometimes it's bad. And we have, I see the T-cell response as a backup. So if you are a dengue vaccine manufacturer, would you want to rely on a vaccine that elicits just the antibody response? The answer should be no. Yeah. Yes?
Is there any way to simulate the processes and so forth with digital simulation to avoid all the analog solutions to the problem?
So the problem is we don't understand enough about the antibody response to this virus. We don't know what are the molecular features of the good antibody response versus the bad antibody response. We don't even know what the antibody response in children versus the elderly, the primary infection versus secondary, the infants versus the older children. We don't know. We don't know any of that. So it's going to take several decades of research, but you can imagine we need that information so that we can design appropriate vaccines. So people like my colleagues and myself were very invested in trying to understand what are the molecular pathways of this antibody response, both in the good setting and the bad setting, what genes are controlling these antibody responses, and forget what genes are controlling. What are these antibody responses? We don't really know. What people are doing right now, we're about 30 years behind the HIV field. So people are going and cloning monoclonal antibodies. What that means is they're taking cells from the human blood of these infected patients and getting out B cells. B cells are the ones that make antibodies, and trying to see what kind of antibody response each individual B cell clones are making. And you can imagine it's a huge fishing expedition. It needs to be done, yes, but we need these kinds of experiments in a massive scale because your antibody response will be completely different from mine. And so we need to do these kinds of experiments, studies in multiple countries. You can imagine, I already told you, the Caucasians and the Asians are the susceptible. The African genotype, they're not. So we need to be studying the African immune response so that we can, but none of that is going. So it's very important to do the basic investigation, both using human patient samples, and I hope I have highlighted the value of using an animal model, how the field suffered for decades, not knowing if this hypothesis could even be true or not.
Is the elevator the way down?
Yes.
Maybe this problem is bigger than climate warming.
Well, climate warming affects the mosquitoes, and the mosquitoes are the big problem here. I think I see them as all related. And so I'm going to tell you a little personal side of my dengue story, which is I'm from Nepal. I was originally born and raised there. And I got into dengue because I wanted to work on a problem that affects that part of the world, on developing countries. And what's really, I don't know if it's good or bad, I'm ambivalent about it now, is dengue is now endemic in Nepal. So I'm in a unique situation where I can really help figure out what is really happening. We know because dengue just appeared in Nepal in the past 5 years, we know nothing. We don't even know what kind of viral genotypes, what kind of viral strains are circulating, how these people are getting. And Nepal is an unusual country because it's not a temperate country. Most people think of the Himalayas, the mountains, right? So this is the impact of global warming, which is these mosquitoes are moving into habitats where they shouldn't be in a country like Nepal. And we don't know, maybe the virus is changing too, but we don't know. We need to do the research. But somehow this combination of this climate change, globalization, is moving this virus into areas where they shouldn't be. They've never been. And now, so in a country like Nepal, if you start to have severe disease, so far the number of severe cases have been low, but you can imagine, the point of my showing chikungunya is that these infectious diseases are very explosive in nature. You can't predict how they're going to behave. And so that's worrisome. And you can imagine I'm trying to set up studies back in Nepal.
Okay. Sujan, thank you very much.